So our first speaker this afternoon is Phil Tailby, who's senior wheat breeder at Limagrain. Phil is not from a farming family. He's got a summer job working as a harvest on trial plots and thought, this is a life for me. He went to Birmingham University and read plant science and then did a master's at John Innes in plants and plant genetics. He's going to talk to us about wheat breeding. In 2020, there were 761 million tonnes of wheat grown in the world on 220 million hectares. Since 1960, world production has doubled. A lot of that is due to Phil and his colleagues in plant breeding.
Thank you very much, and thank you very much for the invite to come and talk today. Very much like Roger, I found out yesterday about the theme being the challenge of net zero. And
your colleague Ron for that, he was told.
Yeah, first of all, apologies, you only have me today. You were going to get Ron as well. I'm sure you all were looking forward to listening to, but unfortunately Ron couldn't make it, so he's stuck with just me. And I also found out that yesterday I was on the graveyard shift, so that's a win-win. Okay, so I'll talk to you today. I was asked to talk about modern breeding techniques. So I'll talk about how we do it down in Woolpit in Suffolk. Well, I'll make a start. Okay, so I'll talk to you, a little bit about our objectives. Obviously, some of the external factors, I'll go over that quite quickly because you all know about those. A little bit more in depth about the breeding timeline, and then a little insight into some of the breeding technologies we use. So the crosses we make today, actually down in Woolpit, they'll be doing that today. They'll be making the crosses which we will hopefully see in the marketplace in about 8 years. So we need to predict the future to a certain extent. So the changes that we're going to see, we need to help address these problems, all these challenges. So we know productivity needs to increase, yield consistency and quality. Consistency is vitally important. Varieties we know need to be resilient. We need flexible sowing windows. They need to be marketable, and of course, we need to produce high-quality varietal advice. This is where I would point to Ron, but he's not here. At the end of the day, our grain, our products need to find an end user. So we're very focused on how the varieties we are producing can meet those end user requirements. So, the external factors we all know about. The environment, changing technologies, and of course, a very tricky one being legislation. So we don't know what is going to change. We can make some modelling, we can make some predictions, or we can make some guesses, basically. And we know that some things may well stay the same. There's a high chance we're going to have to put the wheat in the ground to get it to grow, and there's going to be sun, and there's going to be water, and there'll be nutrients involved. Now, this is a very complex matter. Fits in nicely, this probably one slide fits in nicely with the theme. But there are climate change, it is happening. Weather events are going to become more erratic, water availability, temperatures will all become more and more challenging, and this will affect the way that wheat grows in these environments. Now, a big part of my talk will be talking about breeding technologies. The fantastic tools we have available now, and there are more tools available which I'm sure you're all aware of. I'll touch on a few very lightly, but we have very powerful tools we are using at the moment, and we're starting to see the effects, how these are changing the shape and the balance of our breeding program, and we're starting to see some of the output of this both in our trials and coming into official trials as well. Obviously, you guys are way better placed than me to understand the implications of on-farm technology. I mean, your combines will look very much like the opening picture. My combines are a lot smaller. The tricky one. We do not know what will happen. It will probably change. In England, we like to change our prime ministers very regularly. So we don't know what's going to happen in the future. But as a breeding company, we are multinational, and we have the kind of flexibility of working in all territories around the world. So we can explore lots of different avenues and technologies. And of course, pesticide withdrawals. You can talk to Roger about the different regulations involved in nitrogen and nitrogen applications. And then there are changes in how the end users use the wheat that we produce. So, now a little bit into the breeding timeline. It all starts with crossing, and this is what the crossing program looks like at the moment in Woolpit today. So basically, when you're looking at crossing, you're looking at combining genetics, and you need diversity. Now, diversity just means different. You need useful diversity. So the idea is we design our crosses to capture all the diversity, the useful diversity we need, and then it filters into our breeding process. Now, one of the first big steps with our improvements and use of technology is we genotype all of our material in our breeding program now. So we spend a lot more money on genotyping today than we did 10, 5, or 20 years ago. So this allows us to combine genotyping information with phenotypic information to have very powerful models that allow us to understand the true potential of the crosses we can make. So, we have the term idiotypes, which is basically it's a biological model of the morphological and physiological traits in the wheat plant. So it's the blueprint for what we're looking at producing for 8 or so years in the future. We can specify that a little bit more by that's the major genes handled by informative markers, and the quantitative traits, so your yield, height, earliness, lodging, so lots of small genes, small effects, interacting with each other and interacting in the environment. So very complex. You can't handle them with just a single marker. It's not that nice. So we have major genes, which is often like disease resistance and quality traits. And then we have the quantitative traits. So we genotype everything. So we genotype our parents, so we know what they have. Then we can run cross predictions. So again, if Ron was in the room, I would point to Ron and say, 20 years ago, if you said to Ron, we will be able to tell you with a relatively respectable level of certainty which crosses to make and which crosses not to make by predicting the potential yield, predicted quality of those crosses. He'd been very, very excited. And then, once we've made the cross predictions and decided what crosses we want to make, we then select the most appropriate and efficient scheme to bring one of those products to market. So, don't worry, for those in Limagrain, this is all anonymized and this is all simulated data. But this is something that, just an example of where we're looking at what we have here. So if I find my pointer, here we go. So we have cross of these two individuals, doesn't matter what they are. And then we have a prediction of the yield potential of this cross. And then we have various different quality traits, barometer traits, height and septoria, and yellow rust, and all the other traits. These spreadsheets go on for miles and miles and miles and are very, very complex. But what the point here is to show you that by genotyping and having plenty of markers and resistance genes to look at, we can then decide which crosses we can make to hit those idiotypes. So if we say we need at least 5 septoria genes in combination, we can see. Obviously, there are some things we don't know. There are resistances we haven't mapped and characterized and don't have markers for. But where we do, we can select the crosses which we can put these combinations together. Same with yellow rust, same with quality traits, and so on and so on and so on. One of the tools we have are double haploids. And these have been around for at least 20 years. So basically, it's a way of producing breeding material that is fixed and in a faster way than letting it go through generation to generation. So instead, you create your F1 plant and you emasculate for crossing. Instead of using wheat pollen, you add maize pollen instead. Triggers the formation of an embryo, and then you have to double it using a chemical. They're grown on a medium here like this. These are transplanted out into soil, into a glass house, and then we end up having fully formed, normal-looking wheat plants, which are fixed for those traits which we're interested in. The other option, single seed descent. So this is grown in growth rooms, controlled environment growth rooms, with 2 to 3 cycles per year. So we're speeding up the process again. Now we always like to get a little bit more bang for our buck. So just like the double haploids, with the SSD, we take leaf samples. So if you want to see, it used to be called a muddy boots breeder. If you want to see what a modern breeder is, they'll have green fingertips because you rip a little leaf, fold it up, put it in a plate, and then for us, we send it off to France to our robotics lab. We do all the genotyping, and we get spreadsheets back. So as I said, they're sent to France. So this is what it looks like in real terms. So if we have, I have a pointer. So if we say that this is one cross here, and there are maybe 500 individuals in that population there, they have been genotyped, and we have selected the ones that have the combinations of genes we're interested in: yellow rust, brown rust, mildew, septoria, quality traits. And then we also select from within those ones the lines that are higher yielding, better specific weight, the correct height, and everything like that. So the more genes you are selecting for, the greater the size of the population you need to get all those combinations together. It's just probability. So as you can see, in this, say this cross here, we may have only been selecting for a number of traits. In this population here, we were selecting for more. So there's fewer plants that have those combinations you're interested in. Can't get rid of that flying Frenchman there. So this is a way of speeding up our breeding process. This has been going on for a few years, but now we're improving it, we are augmenting it with our genotyping, our marker-assisted selection from major genes, and our genomic selection. And finally, you need a good size ear to go and put it out in the field. Now we have lots of fields, some with trials in, all breeders like photos of trials, and breeding nurseries. And so each one of these lines here, about 1 metre long, and contains at least 10 or so plants. And so this field, 10 years ago, may have had 100,000 potential breeding lines. Maybe 50% of which, as breeders on our first selection, would have walked through and just discarded them because they died of yellow rust. But now with the work we're doing in the growth rooms, this field is slightly smaller. There's maybe 50,000 lines in there, but we're not throwing them out anywhere near 50% just due to yellow rust or brown rust or septoria. So it's a much more efficient way of doing your selections. That's actually me there. My hair was shorter and I was a lot younger, but we use small tractors and we put material out in the field. We can do some very fancy things in growth rooms, but we still need to have material out in the field, whether to multiply up or phenotype look at, basically. So maybe a few years ago, it may would have been looking to see if your material was resistant, or the parents of the crosses you want to make are resistant. Now it's more along the lines of we are monitoring resistance genes combinations. So currently this year we have at least 135 different unique yellow rust combinations we are monitoring to see that they work. So we find resistance, map it, combine it, monitor it. And again, we still look at plants in the field. Rather than books, we generally use tough pads or your phone. Basically, we collect a lot of data. And we also need to look at the disease. So it's a lovely picture here. It's very clear. Very, very, very dirty. Very, very clean. And so now, not only we're selecting the material that is clean, but we are mapping, as we go, new resistances to feed into the pipeline to get us some more resistances going along. And again, another picture of yield trials. We love pictures of yield trials, especially from drones. But they're critically important. We have a lot of power with our genomic selection tools, which I'll cover in a moment. But without excellent trials, we don't provide excellent data to go into our models. And we all know rubbish in, rubbish out. So trials are critically important, and I'm sure Adam will definitely be nodding his head, very much so here. They are vital. So look after your trials officers. Really? Where are you? Where are you? Couple of quid, yeah? Thanks. Okay. So again, yeah, here's one of our dinky little combines. They have onboard weighing. So we either combine little plots, keep the seed for further analysis, and further generations, or locations. Or we just weigh and go, we call it, just to get the data. Okay. Familiar map of where wheat is grown in the UK. And as you know, especially when talking to this room in Scotland, most of the plant breeding is based around here. Sorry about that. And we know that we're breeding for the whole of the UK. So we need to look at yield stability in lots of locations. So we have a trial network in lots of locations. And we also have disease nurseries in lots of locations, because we know disease in location A is not the same as diseases in location B. There are different pressures. So again, we need to put out our sentinels, our combinations that we need to check in all of these locations. So it's not just a single area we're looking at. We're looking for lots and lots of environments. Okay. Now on to the breeding technology. How are we doing for time? We're okay. Okay, so a little more into those DNA markers we were talking about. So this enabled us to identify the genes that control a trait, or specifically a marker somewhere near those genes or that gene, so we can follow it in our marker-assisted selection, select those right plants. So the DNA markers are like an address we follow. They're the science. And then we've got the gene, which controls the trait of interest. So you've got your gene that gives resistance to brown rust. And the marker may be nearby. A perfect marker is within the gene. So you know you've got them. So that's why we need to continue to monitor and check, because you can get recombinations. You might just have the marker without the gene. So it's not, unless you have a perfect marker, you still need to keep refining and doing the work to make sure you're making these selections, because you're doing it very early and you're basing a lot on these markers. They're very, very important. So the improvements over the last 20 years, a lot has been driven by the fact that we can now genotype, so the cost per marker point has decreased enormously. It's very much like the chips in your computer. So you have greater processing power and it's effectively cheaper. So same with genotyping. We can now genotype high density all of our breeding program. 5 years ago, we couldn't do that. 10 years ago, we certainly couldn't do that. I'm sure in the future, whether sequencing of individuals, if it's an advantage, could well be an option. Now, the really fun stuff. Genomic selection. It's been done in animals for quite a while now, so I'm sure you're familiar with the concept. But I like talking about it, so I'm going to talk about it some more. We're not looking at just the individual genes, we're looking at lots of small genes. We're looking at the whole genome here. Okay, so we genotype pretty much everything. So you've got that information. And then we measure the traits, everything. Yield, height, specific weight. You combine high quality genotyping, high quality phenotypic data, again, trials officers, brilliant. And then you can use high power statistics, create models which allow you to predict with a decent level of accuracy how something will perform. So, and then you can predict the material you haven't seen in the field, how they will perform. So again, you make your selections in your growth room. Now, one of the big advantages and strengths of this is we've phenotyped material over lots of years and in lots of locations. So when we have a prediction for a score for, say, yield, that is based on lots of years and lots of locations. So a lot of the stability and a lot of the, we call G by E, genotype by environment interaction. So you're getting a lot more power there. So you're actually seeing how potentially this prediction, the potential of this new variety will perform in lots and lots of locations and in lots and lots of environments. So not just testing it in a couple of years, releasing it. We're having a lot more information over many years and many environments. And now the environment's changing more and more. This adds a lot more power to our selections. The breeder's equation, which we live by as breeders, if you can go faster, you can get more genetic gain, basically. So, a few years ago, to identify a good parent, you'd have had to make a cross, put it in a field, try and kill it with disease. Select the cleaner ones, put them into yield trial. See the ones that yield quite well, test a bit more in a few more environments, and then you might identify that's a potential parent. So you can feed that back in and around and around. Now, when we've made the cross, we have our material in our growth room. These are the winners we're going to pick. We can also see the potential parents at that stage. So we've shaved 3 or 4 years off. So we can increase our genetic gain. The time it takes to produce from cross to a variety, similar length it was 5 or 10 years ago. But identifying parents, and therefore driving the genetic gain, is faster. So that's one of the big advantages. So, a live example of how we use cross prediction. Don't worry, I know it's after lunch, and these graphs are nowhere near as complicated as Roger's. I'm a simple breeder, he's a scientist. So, 260 potential parents. So you've got about, in blue, 30,000 potential crosses. So here we got specific weight and yield. Okay, so the potential of those crosses. About 4,000 of those in red. We could get those combinations of yellow rust, septoria, brown rust, mildew resistance. Those were the ones that had enough potential to hit those idiotypes we're looking for. And in fact, we made about 400 of those because they're the ones that tick the boxes for high enough yield, the right height, not too late, not lodging, and all the other quantitative traits. So from a potential 30,000-ish crosses, with a lot more accuracy than we could 20 years ago. We can narrow it down to about 400. Because we can't do everything. No matter how much money my French bosses would like to reinvest in breeding, we can't invest billions and billions of pounds. So we have to be quite efficient in the way we do it. And this is the strategy we use. Now, gene editing. You've heard lots and lots about gene editing, the CRISPR-Cas9 system, but basically the Cas9 is an enzyme that basically acts as scissors and cuts the DNA. You've got a piece of guide RNA that basically you've designed to find the gene you want to edit, and it goes in, and depending on what you want for that gene, you can make adjustments. We're a multinational breeding company. We work in territories all over the world, and some places are amenable to GM and gene editing. We know in the UK, currently, it's not. But we have platforms to use these technologies if and when they come on stream. So there are potential uses for the future. The diversity part I talked about earlier, you could possibly, you can, create your own diversity in a very controlled and targeted way. So there's a potential there. But currently, it's not a tool we have in our arsenal. Okay. How are we doing for time? 5 minutes. Well, I mean, I can go into a little bit about some traits, but I don't think that will keep with the theme of the topic of net zero. So, are there any questions?
Okay, we've got time for...
Yeah, we'll have some questions if you like.
Time for a couple of questions. We're going to have a forum after Paul's spoken, so just if there's anybody...
Anyone awake after lunch and that? Friendly comments. Thank
There's
you.
an elephant in the room, Phil. Soft wheats in Scotland fell apart last year with Septoria.
Mm-hmm.
How long is it going to take you or your fellow breeders to come up with robust varieties that are going to allow us to continue growing soft wheats but without being swamped by Septoria? I think that needs to go further.
Oh, okay.
I think it's feeding
Okay.
back to the...
Okay. Well, so the difference between hard and soft, you could say is one gene, the endosperm texture. And there's a difference between Septoria, robust Septoria resistance, and poor and non-durable Septoria resistance, maybe 2 or 3 genes. So you make your cross, select for the soft material on that one gene, and then make sure you have your combinations of the Septoria resistance you know are effective, and increase and bring more in. So they're in the pipeline and they are coming through. We have a lot more Septoria resistances we know about and markers we can deal with because, for example, we're bringing in new diversity. We're better at genotyping. We can genotype more. And for example, in 2016, we had a really good year for Septoria. Sorry, for breeders, when we have lots, we say that's good. So we can score all our material, and then we can map and identify these resistances we've got on our germplasm, and then we can start manipulating those early doors in our crossing programs, in our growth rooms, to put those stacks and those combinations together. So it's coming through. But as I said, the crosses you've seen we're making today are not going to be ready for, we'll see in the field in 3 years' time. We'll screen in high-pressure locations in Ireland and other locations around the UK to really test these combinations out. So that in the pipeline, and they are coming through. Soft or hard, tall, short, not too late, mildew resistance. It's all there, it's all possible. And we're putting those pieces together. We've got the idiotypes we pick, and that's why we want feedback. If you have specific demands, we can, through our breeding by design strategy, we can match and meet those targets with the efficient tools we have. So we're doing our best, we'll get there.
So answer your question, Adam.
I'm not going to give you a date. There's cameras, there's people making notes.
One more question. Thank you. Yes, James. Microphone just coming.
Thanks, Phil. Interesting talk. I just wondered.
There's something right to your mouth.
I I just wondered if what are we missing out on with you not being able to use gene editing in the UK currently? Are there significant things we're missing out on, or is it just a handy thing for you to have?
Good question.
Repeat the question, please. We can't hear. Said, what are we missing out on not having gene editing in the UK? Is it a handy tool for Phil, just, or are we missing out on serious potential benefits?
Well, I think anywhere where you can make the targeted diversity you need, you can improve your potential resistances, whether it be for new diseases or new pests coming through. The question is like the speed aspect. So if a new insect pest comes along, starts attacking wheat, the usual approach would be have a look round, find is there enough or any diversity we can use in elite wheat. So we'll have a look round, see if there's anything, put it out, subject it. If it all dies, no, that's not good. Then you go and look a bit further out, so you start looking at this pre-breeding material and slightly crazy looking wheats. And if there's some resistance in there, you need to identify it, and then you need to bring it into your crossing programs, and then you need to follow it for your breeding programs, and then you can then start putting it into your more elite materials. So that's quite slow. But if you can find a resistance, you have a gene that you can identify the gene, which can provide resistance in something else, and then look kind of in silico bioinformatically, is there something very similar already there in wheat, and edit it, you can shorten that timeline significantly. But it's complicated. It's a very powerful tool, potentially, and we're learning how to use it. We're learning as we go. I say we, I mean colleagues in other territories in the Limagrain Empire. But yes, it's potentially a very huge tool there, but it comes at cost.
Right, thank you very much, Phil. A really good, in-depth talk about wheat from beginning to end. So can we all thank Phil, please?